Hello, and welcome to another episode of Thoroughly Thriving, where we're putting human back in human capital solutions. On today's episode, we're going to hear from an industry leader, where they're sharing their thoughts, feedback, and perspective on what's happening in today's world of work. Well, let's stay tuned and perk our ears up for another exciting episode. Well, welcome to another episode of Thoroughly Thriving. And do we have an amazing guest with us today? Today, we have on the show Greg Opatek. And Greg is based here in San Diego, sunny San Diego. And, as the leaves are changing, these are the times where, the sunshine is still very, very bright in San Diego. And hopefully where you are based as well. But on today's show, we're going to hear from Greg Opatek. Greg is a senior life science leader, and he currently is the head of precision and translational medicine at a Boston-based biotech company called Affinity. But Greg's experience is extremely extensive. He has made so many valuable inputs into global health, into really innovating new medicines that certainly are going to extend and provide many solutions as it pertains to global health. Greg's career has spanned across many decades. He's worked for large pharma, he's worked for biotechs, and he's got a really good perspective on not only translational and precision medicine, but it's been able to work cross-collaboratively with so many other functions across clinical development as well. So we're excited to have Greg on the show today. We're going to understand and learn some key insights from his experience, and I couldn't be more excited. Sean, what are your thoughts today?
Greg, welcome so glad to have you. And yeah, I've been personally, following a lot of the great work that Greg's been doing over the years. So excited to have him join us today and really get into this conversation.
Thank you very much, Darren. Sean, it's great to be with you guys. I'm super excited and a big fan of your show.
Awesome. Well, as we started to think about your background, we typically have a lot of leaders, innovators, and KOLs that are focused predominantly in clinical development and cross-clinical research. And Greg, when we met earlier this year, you had just a passion that was not common. The way that you talked about not only the company you're at, but your role within the company was a bit uncommon. And I've used the word, but I'll use it again. You just exuded passion. But as we kind of take a step back, I'm curious for myself, for Sean, I'll speak on Sean's behalf, and a lot of our listeners, can you explain to us what a translational medicine specialist does within the confines of a pharma and biotech company?
Sure, Darren. So the role generally exists at the interface between research, clin dev, clin ops, maybe with a little biometrics thrown in there. We spend our times trying to build bridges and fill gaps that naturally occur in organizations. The majority of our time is spent trying to figure out which biomarkers need to be assayed, should be assayed, where and how to assay them. And so it requires a translational scientist to spend a lot of time understanding what's coming out of the research organization and heading into the clinic. A lot of time with the ClinOps folks figuring out things like schedule of assessments and kitting, as well as central labs and referral labs. And then with the clin dev and the biometrics folks, understanding questions like what is a response, what's a partial response, how can we understand an exposure-response relationship? So it even touches on clinical pharmacology as well. But through it all, the important thing to remember is, like you alluded to earlier, why are we here? Why are we doing it? And it's really about the patients and about improving human health. And as long as that's our guiding North Star, then oftentimes you can take work out of the system and better prioritize your day-to-day activities.
Thank you, Greg. Can you share with our listeners how you identified getting into this space specifically across drug development ultimately. Was it something where you knew coming out of getting your master's and your doctorates that you wanted to go down this route and why?
Yeah, so I started out after my PhD working at Bristol Myers Squibb in the Department of Applied Genomics under Elliot Siegel. And he was an incredible leader, dynamic visionary, and he had come out of biotech and he had just come into pharma. And I think it was under his leadership and mentorship that it really spoke to me, which is we've got a mission to try to bring innovation to big pharma. And I'll give full credit to Elliot, who taught me back in the day. Big Pharma really does late stage development, regulatory approvals, manufacturing, and marketing well. That's the sweet spot of Big Pharma. But at the time, to answer your question, when I got into it, let's be honest, Big Pharma's research and discovery engines weren't all that productive. So starting then, I realized, what can I do? How can I contribute? How can I bring my skills to bear? And it really became apparent to me that a lot of the research going on in academics, as well as in big pharma at the time, was research for the sake of research. Publishing papers makes sense. Improving knowledge and wisdom makes sense. But it was mind-boggling to me back at the time how few of those discoveries ever made it into the clinic. And so that was the critical inflection point in my own career where I saw myself in the Department of Applied Genomics being able, with new and modern technologies, to look at PK-PD relationships in patients, in subjects, in real time, and understand, were we inhibiting the enzyme? Were we filling the receptor? And how is that changing the course of human health? And once I got into the clinic, started running assays on human samples, everything else pretty much fell into place. Because when you're doing that, you're at the pointy end of the spear. And when you're at the pointy end of the spear, the job is never dull or boring.
Well put. Well said.
Great. I think that's so well said in terms of, I think sometimes where people in our industry get a little worn out is when they do get away from that place of really being the tip of the spear, and being involved in things that excite them. I can't tell you how many people I've talked to in my career who have such enthusiasm for the work, but it begins to grow a little weary when they do get further and further from that tip of the spear and into the load of the work that has to be done. You've made some great growth steps in your career, and you've moved kind of back and forth between different organizations. What would you say would have been some of the most critical decision points in your career that have led you to the place where you are now?
Yeah, when I left, let's say big pharma to go to biotech, that was clearly a critical decision point, and that was really driven by that same story I told you earlier, which that had happened 15 years earlier. So I worked in big pharma for 15 years and realized I got to the point where I had become good at the bureaucracy and navigating the Byzantine labyrinth that is big pharma, but I didn't get excited about, hey, I just navigated that labyrinth, right? And so my critical decision point was leaving that behind, moving to San Diego, working in biotech, and wow, things got fast quickly. Decision making, governance, go no go decisions just happened at the speed of light in biotech. And I think honestly, that's what you see over the last 20 years. That you see big pharma realizing that innovation comes from biotech and maybe not so much within their own organizations. And so look at Bayer Leaps, you could look at J&J Labs. There's lots of examples of big pharma fostering, catalyzing little biotech, little ideas to take root, to move to POC. And then guess what happens? Big pharma comes in and buys the little biotech. And I think there's a growing realization that it's really hard to do that, IND enabling talks, first in human, clinical POC in the confines of a big organization where you'll have 20 different department heads, any one of whom has veto authority over that program. Any one of whom can say, I have some concerns. I think we need another year to study this. And, unfortunately, that really takes a toll on the patients as well as the scientists within Big Pharma. But also, if you talk to folks like Deloitte, it takes a toll on the ROI within Big Pharma itself.
Wow. That's a great perspective to think about, Greg. And, the innovation that we've seen even in the last 10 or 15 years, I wonder how many scientists who did research over the previous 20 or 30 ever thought any of that was possible. What has been, maybe one or two, research or discovery element things you've been involved with? I don't need you to get too much into the weeds, but, that you've been really just most marked by in your scientific career.
Oh, sure. So that's an easy question because about 20 years ago, a colleague of mine who was working at the bench next to me discovered a new drug target called Bruton's tyrosine kinase. And I remember that gentleman beating his head against the wall, going to department head after department head, going to governance after governance, trying to convince them to put together a lead ID program around what's now known as BTK. And it was incredibly educational for me to see him get his soul crushed because there was never enough data to convince governance to enable the BTK program to go forward. I mean, I would sit in governance meetings and watch his presentation, and they would say things like, how can you be sure this will work in the clinic? Well, that's a gotcha question. You'll never be sure until you do the experiment. And if big pharma and governance committees want to hold every discovery researcher to that level, I guarantee you one thing, they will never take a new target into the clinic because there's no such thing as a guarantee before you start lead ID chemistry.
Well, I'm just reflecting back. I'm imagining how challenging that could have been and was for your colleague. Well, you're exactly right. Look, I think we all can identify that our crystal ball is cloudy on the best day, and not until it gets into the clinic, until we start doing the experiment, as you put it, Greg. That's when we're going to start to really understand if it's got some certainly efficacy and it's got some legs, for a lack of a better term. So, Greg, now you're in cell and gene therapy. I'm curious, given your extensive and expansive career, um, now working in cell and gene, how different is it from working with a small molecule or even a monoclonal antibody study program focused company? Can you share with our audience some of the key differences, some of the nuances that you've been able to really see firsthand?
Yeah. When you're in cell and gene therapy, not always, but 80% of the time, you're doing single arm open label studies. And let me tell you, doing a single arm open label study is a whole heck of a lot different from placebo controlled randomized clinical studies. And it's both good and bad. So let's start with the good. So when you're doing a single arm open label study, everybody's getting your drug product. And that's, takes all the mystery out of it. But it also raises the bar in terms of the turnaround time of data generation. So when you're a translational scientist, you need to be working closely with your ClinOps group, your ClinDev group, maybe Med Affairs, but definitely the clinical sites to ensure that they're collecting the right samples from the subjects at the right time, doing the right things with those samples, which is generally sending them off to a central lab. And then generally the central lab will run whichever assays you dictate that they can. Assays that they can't run, they generally outsource or subcontract to a referral lab. But imagine that process because you'll have a flood, a tsunami of data coming back to you in real time. And so when you work in cell and gene therapy, there's very few nights and weekends or holidays where there isn't somebody asking you, have you seen the day 15 data yet? What's the Cmax looking like? Have they peaked yet? Have you calculated the AUC day 1 through 29? All those questions are common and par for the course in cell and gene therapy. But only rarely do you see that when you're working on a small molecule or a monoclonal for that matter. Generally, you'll have an unblinded clinical pharmacologist, and you'll be generally running some values, but it's in a much more batched manner. So every month or every quarter, you'll collect a bunch of samples and you'll assay them, and then you'll look at that data and analyze that data, blinded or unblinded, in a very prescriptive way, which means you can plan around it, right? So, the beauty, though, of being a translational scientist when you're doing randomized placebo-controlled studies is, again, pro and a con, is oftentimes you're privy to data that the rest of the organization isn't privy to. And sometimes it gets awkward because sometimes you could potentially be unblinded to active versus placebo, and that would make you in possession of material information. And I can just imagine all your listeners who are lawyers or finance folks getting, sweaty right about now as I say that. But that's one of the upsides of being a translational medicine scientist is you get to peek at what's going on in close to real time, even in placebo-controlled studies. And again, that brings us back to what we talked about at the top of your show, which is it really reminds you in the translational medicine space that you're at the pointy end of the spear. What you're doing matters. And there are real patients, and you're getting data from them in real time. And sometimes that can be sad because you see their disease progressing, right? You may even see their disease progressing before they see it because you may be in receipt of diagnostic data as part of the clinical study that their bedside oncologist hasn't yet shared with them.
Thank you, Greg. To look at that and understand from a very high level the nuances and the intricacies and how they differ certainly are very apparent. And you're right. It seems that collaboration, working cross-functionally with your teams, are of the utmost importance. Greg, I'm curious too. Given your background and certainly the different organizations that you've worked with and for, and how you've progressed through your career, this is a two-part question. What's the best piece of advice that you've received either from a peer or mentor throughout your career? And conversely, um, if you can recall this, when, what piece of advice would you give for someone looking to get into translational?
Excellent. So, best piece of advice ever goes back a couple of decades to a woman by the name of Nikki Katz. And I haven't kept in touch with her, so I don't know if she's still active in the industry, but if she's out there listening to this show, big ol' hello from Greg Opatek. What she told me one day in her office years ago was, this is an attrition business, Greg. And what that means is the earlier you are in the pipeline, the easier the job is to do. Because when you're in the target ID space, you know that maybe 1 out of 100 targets will ever see the light of day, let alone get FDA approval. When you're at the first in human, it's probably 1 in 10 if you're lucky. And when you're at phase 3, it's 1 in 2. And I sat there as a young scientist listening to Nikki, and I said, so how do you put that into practice? And she says, oh, easy, Greg. I say no to everything. And she says, she waited, she paused, and then she said, and I'm right more than 99 times out of 100. So by her teaching me that early on, rather than me going through my career, naively, I'd like to think that thanks to her, I went through my career a little bit more savvy, a little bit more like we talked about earlier. When I go to a governance meeting, look around the room and try to figure out which of those folks sitting on the governance committee are going through the motions because they know the answer. They know at the end of the meeting, they'll simply say no and always be right. And the one time they're wrong, getting back to the BTK story, they can always blame the messenger. They can say the person who presented that 5 years ago when we shot it down didn't adequately convey the upside of the BTK program. It's not my fault. It was the presenter's fault. And I think if you take that thought process into everything you do in pharma and biotech, you'll have a lot more success, and you'll definitely understand the position that a lot of the folks come from. And back to your other half of the question. So what piece of advice would I give to somebody going into translational science? Look in the mirror, understand who you are. Understand if you always need to be right, or if it's more important that what you're championing, let's say an asset or a biomarker or a clinical study, gets the green light. Because as Lyndon Johnson said 60 years ago, you can get anything done in this town as long as you're willing to take the blame and give away the credit. And the same holds true in translational science.
Darren, I'm stealing that one. I'm going to write that one down. I think that's great. Greg, I got one final question on my end that I would love to hear your thoughts on. You've worked on a lot of innovative programs over the years. What are you most excited about the next 5 to 10 years in our industry? What are you looking most forward to? What excites you? What do you think are some of the cutting edge breakthroughs that may or may not be coming, in the next few years that you're keeping an eye on?
Yeah, I think we're getting a lot closer to moving the needle on solid tumors in oncology than we have ever been in the past. And I give a lot of credit to the folks that came before us, especially at the NCI, who went forward in heme malignancies like DLBCL and multiple myeloma with the whole CAR T revolution. They demonstrated that you can really get durable, long-lasting cures in heme malignancies. You can really bring a cell therapy to market, and you can really make it a successful drug that transforms the lives of patients, providers, and even payers. And I think right now we're on the cusp of doing that in solid tumors. So I'm excited and optimistic and think there will be lots of good news coming from the field in the near future.
I don't know about you, Sean. I've learned a lot today. I've definitely learned a lot. Thank you, Greg. This has been enlightening. It's been informative. I also feel equipped. So that's a really good trifecta for me, Sean. I don't know how you feel about it, but I'm sure you can identify.
Yeah, I think we're gonna have to have Greg back. I don't think 25 minutes is enough time to, for us to learn, Darren. But selfishly, maybe we'll have to just take him to lunch or something for our benefit. But it's one of those things. I feel like 25 minutes is not near enough time. I feel like I could listen for another hour.
Well, thanks, guys. I had a great time today, and it's always a pleasure, and I'm always happy to chat.
Sounds great. Well, thank you all for tuning in to listen. We'll definitely put some links in the profile notes here for the episode so that you can follow up with Greg and continue to track his career, some of the exciting work that he's doing. And thank you again for tuning in for another wonderful episode of Thoroughly Thriving, where we continue to focus on winning as one because we work as one, we can win as one. Thank you so much for listening today. Have a great rest of your week.
Bye, everyone.
Thank you for joining us today for another great episode of Thoroughly Thriving. If you enjoyed today's episode, please feel free to share it with those in your world. If you'd like to learn more about the Thorough Group, please go to thoroughgroup.com. And remember that we will all remain thoroughly thriving if we continue to work as one and win as one.